Now let us go for markers of apoptosis or investigation to find out the cell has undergone apoptosis. First thing which I want to mention is dye, which is going to find out annexin 5. In case if that cell is going to emit fluorescence, means that cell has undergone apoptosis. Please note it down. I will inject the dye to the cell, and if that cell emits fluorescence, that means the cell is able to bind with annexin 5. Only if that annexin 5 is binded, a fluorescence will be emitted. That means apoptosis positive, or that cell is undergoing apoptosis. Now let me explain the reason behind this. In the previous slide I mentioned, let this be apoptotic body. So what will be the present in the surface? Please answer. Yes, phosphatidylserine. Annexin 5 be able to bind with phosphatidylserine only if it is outside the cell. Obviously only in apoptosis it occurs. Now let this be normal cell and phosphatidylserine is inside. Now this annexin 5 I am putting, no binding, because annexin 5 can bind only if the phosphatidylserine is outside the membrane, which is present only in apoptosis. Only because of all the caspase activation, that process of apoptosis, that phosphatidylserine will come to the surface. Only if it comes to the surface, binding occurs. Here binding occurs. Here no binding into. So no binding. So this annexin 5 which is going to produce fluorescence, here no fluorescence emission. That means normal cell. In apoptotic cell, the phosphatidylserine is coming outside the cell, and that phosphatidylserine can combine with annexin 5. When I am injecting annexin 5, that will combine with this, and then fluorescence is emitted. If the fluorescence is emitted in that cell is undergoing apoptosis or undergone apoptosis. If that cell is not emitting fluorescence, it is normal cell. So that is one of the, annexin 5 is one of the markers of apoptosis. Please note it down and explain the reason also. Then another marker or test, gel electrophoresis, which can be used to find out whether the cell is undergone necrosis or apoptosis. In apoptosis, step ladder, when I do gel electrophoresis, for example the damaged DNA, if I see the damaged DNA when it has undergone apoptosis, when I examine it in gel, when I examine through gel electrophoresis, step ladder pattern. In case of necrosis, smear shape, smear pattern. So another marker or indicator of apoptosis is when I do gel electrophoresis, in apoptosis it is step ladder pattern, in necrosis it is smear pattern. That point you can note it down. First marker what you saw was annexin 5. In apoptosis, annexin 5 will bind because phosphatidylserine has come outside because of apoptosis. As annexin 5 binds with phosphatidylserine, fluorescence is emitted. That's what fluorescence emitted means, positive, which means cell is undergone apoptosis. Third one is tunnel. I am not going to detail, just I am going to mention the name alone. Tunnel stain is positive in apoptosis, negative in necrosis. So I started with definition of apoptosis. Difference between apoptosis, necrosis. Apoptosis is programmed cell death, suicide. Necrosis is cell murder. In apoptosis, no inflammation, membrane intact. In necrosis, membrane is damaged, inflammation is there. In apoptosis, the cell is completely phagocytized. In necrosis, the cell will spread its infection into surrounding cells. So necrosis is dangerous. Then finally I discussed about markers for apoptosis. Tunnel stain I mentioned, TUNEL. Then very important, annexin 5, the same phosphatidylserine which is present outside in apoptosis, that only can combine with annexin 5. Only annexin 5 combines with phosphatidylserine, fluorescence will be emitted. So that is called as fluorescence test. It will be positive if apoptotic cell is there. In normal cell, annexin 5 cannot combine and no fluorescence is secreted. So hope this video is very useful to you.